So coming to the functional genome annotation, so this would be a bit of interesting, or maybe many of you have heard about all these databases or all these bioinformatic methods that can be used for the functional genome annotation. So the image which is being shown in the first part on the upper side of the slide, which is the gene prediction that has been taken out from the structural genome annotation. So this would be further tested, or this would be further annotated for the functional information that comes to know what specific domains or the motifs are there, which are there to have a specific function, or these can be characterized there, or the search for the orthology to check how much homologous sequences, or how what is the phylogenetic characteristic of that particular genome. As I mentioned, that though we may be the first to sequence the organism, but we may not be the first to sequence the first organism, because we all are being derived from the common ancestor, and we all may have the different homologous sequences. We may have the paralogy, we may have the orthology with us, with the different organisms. So that all has to be searched out. And how these are done? These are done by the different kind of software or the databases which are already being available. As I mentioned you, that is starting from the first time, which is 1965 to 1970 era of Sanger sequencing. A lot of DNA has been sequenced. It has been submitted to the depositories, to the different databases. They all help you to annotate the DNA which you have sequenced. So some of them for the domain specific information, to get the domain specific information, is from the NCBI, from which is conserved DNA databases, Interpro databases, which are from the EMBL European labs. Then the family PFAM, which gives you an idea whether the DNA which is encoding for the protein that is having any kind of protein superfamily kind of structure, or it is there in the form of different domain or the motif which is present there. Then you have the signal or the target peptide which is being there. Then you can study for the phylogenetic relationship with the other proteins, or the homology detection with the other kind of databases, for example UniProt, SwissProt, which are available in the public domain, and one can go for the annotation of the function of the genome at the functional level to decipher which kind of function, which kind of specific domains, families, sites, or they can be involved in any kind of pathways or the biochemical reactions they are which are occurring inside the cell. They all can be there. So I would suggest, if you are interested in reading more for this annotation purpose. So first thing that I would suggest you to go through this paper. This is very beautiful paper, and really helped me when I started looking for the annotations or the gene identification information for my graduate students, to let them know about these information. So moving ahead, coming to the challenges, or the coming to the different points which are there, which actually kind of make some challenge to the persons who are going or who are trying for the genome annotations. So initially, if you can recall, one thing was there, c value paradox. That was the term that I used and tried to explain there, that as the genome complexity or the complexity of the organism it increases, it does not signifies with the c value of or the haploid genome of the organism to which the complexity is being assigned. For example, as I try to convey, that we suppose that humans are the most complex organism to understand the at the DNA level or the genome level. But there are certain other reptiles, or like salamanders are there, that are having the 10 times of the more DNA content as compared to the humans. So, and also, if you can recall, I told you while
How
discussing
many
at the
slides left,
point of
madam? How many more slides?
uh, sir, I think 5 6 slides are remaining. I'll try to wind up
Okay,
as
okay.
early
Because there
as.
is question waiting. There are so many questions waiting
Okay,
in chat. Okay.
okay. So uh, um, So how these DNA sequences are not correlating because of the presence of those repeat sequences, which could be the simple repeats like 8080, and we all know these sequences are there. Then the complex repeats like the transposon elements are there. They are being, they can be DNA transposable element, or they can be retrotransposon which gets incorporated into the DNA sequence. So repeat elements, they possess one of the major challenge for the persons who are trying to annotate the genome. So how these are being how these are being tried to curate it, or how these are being tried to annotate it. There are two very prominent softwares, or there are two prominent modelers which are present. Repeat modeler and masker are there. So what they try to do, they read out the sequences which are being obtained from the genome sequencing, and they try to analyze them. And once they found any kind of repeat sequence, they hide it or they try to mask it. How the masking is done? There can be two ways. One is the soft masking and the other is hard masking. Soft masking is what? Just to convert these repeat sequences. As you can see, you can differentiate the soft you can differentiate on the nucleotide sequence which has been put here. So soft masking is what? When you have some kind of repeat sequence, they are being converted into small cases, because in most of the cases we are aware of that the DNA sequence or the nucleotide sequence, or even in case of protein sequences, if we are putting their sequences, then that is maximum in case of capital caps, they are in capital letter, whereas the mass sequences, they are just being converted in the form of small letters. Whereas the hard masking is what? When you have the repeat sequence, those repeat sequences are converted into just the letter N. Then one can have an idea that these are the repetitive sequence, and one can try to take the part one can try to make the partition between the exon, or between the exon and those repetitive sequence. That can be intervening sequence, that can be repetitive sequence, that can be anything, transposable element or the simple repeats. So when there is a long chain of these repetitive sequence, these repeat masking, it provides a very vital tool for those bioinformaticians to annotate the genome data, to get the idea whether they are repetitive or not, or to further analyze them. Now, there are some, as I earlier mentioned, also you cannot separate the gene identification tools from the genome annotation, because ultimately in genome annotation you are trying to identify the locus of the gene, you are trying to identify it at the structural level, you are trying to identify it at the functional level. So ultimately these gene identification tools, what they do, they ultimately provide you the functional gene, converting or deciphering the genome in the form of the functional gene, introns, exons, splitting site, regulatory sites, gene encoding proteins, different motifs, ESTs that are present on that DNA sequence. So all of them are there. So it is basically for the determination of the function or its product from the functional gene. So earlier also we have seen the genome annotation, ab initio predictions, and the evidence based prediction. Evidence based prediction, what they use, they take out the information already available in the form of the databases. You just go for the one of the very easiest way to look for some evidence based prediction is just by taking the DNA sequence, go for the BLAST, basic local alignment search tool, available on the NCBI repository, very easy to use. Just put your DNA sequence in the faster format, you may get a number of sequences of the DNA sequences which may be having some kind of homology to the sequence that you have submitted for the blasting of that DNA. So those are the evidence-based predictions. Then you have the ab initio predictions. What these ab initio predictions are, they are basically based on the two kinds of information. One is signal content, signal sensors and content sensors. Signal sensors are what? These are the start, stop codon, splicing site, branch point, where the DNA is getting bifurcation. Then the content sensors, like if you have the coding region present in that DNA, or the non-coding region is present in that DNA. So all those can be checked through the ab initio predictions. These are some of the tools which can be used For I have just limited these tools to the three or four, or probably these are the five identification tools that are being shown here. Otherwise, you have a lot number of the DNA databases or lot number of the softwares packages are there which can help you to identify a particular gene present on the DNA sequence. Some of them which I mentioned here, these are the ORF finders, GRAIL, promoter, which can help you to identify a particular transcription start sites on the DNA sequence, which can be bound to the Pol2 promoters. You have gene scan, which can help you to identify the gene structure on the genomic DNA of the various organism. It can give you an idea about the exon intron structure of the gene. They are present there. As far as the GRAIL is concerned, it can help you to identify promoter sequences, polyase sequences, CPG islands on the prokaryotic cells. Then, CPG islands are which are present there. Drosophila genome, E. coli genome. These are the can be deciphered from the GRAIL. Then you have gene ID, which is one of the most known, or what I say, one of the oldest kind of depository or the database which can help you to have an idea or get it identify, or the gene can be identified with the help of this gene ID. So these are some of the identification tools are there. Then what I said to you earlier, that the easiest one for the gene identification tool, if you have the DNA sequence with you, just go for the blast. This will give you the first idea what your DNA sequence, what your protein sequence, it can perform at the primary level, give you the basic idea. There are the different kinds of blast which are present on the NCBI. If you go to the website of NCBI, blast P, N, X, N, T blast N, T blast X, these are the different programs. Input data, you can have the protein. If you are going for the blast P, then it will you should have the protein sequence or the amino acid sequence which will be searching against the protein. Similarly for the blast N, X, so these are the different input data that can be checked for the different forms of the biomolecules, whether it can be proteins, it can be nucleotides, or these are even the translated versions. So they can be also checked. So there are some of the sophisticated blasts which are present there, 5 blasts. When you are not hitting any kind of, when it says that your protein is not getting any information, or it is not correlating with the earlier existing protein, you can go for this side blast and 5 blast. They are there. They are more sophisticated softwares or the online tools which are available. You can check for the presence of different structural or you can say functional annotations of the protein or the DNA. You can go through with them. So, in the end, why this topic genome annotation and gene identification? It is important. So, this slide, these three discoveries. The first one, if you read out, Craig Venter, it creates synthetic life form. This is the first report in 2009, which was from the group of Craig Venter. So what they have done, they have created the first synthetic life form, which was mycoplasma. Mycoplasma, if you can remember, it's a kind of eubacteria which is existing. So this was the first report that they have used a life form, completely made it from the synthetic methods. How they could do that? They have the information from the genome projects, that what kind of DNA or how much DNA is important if you want to synthesize cell inside a lab. So this was the first report for the synthetic life form which was being there in year 2010. This is the report. Scientists create simple synthetic method that can grow and divide normally. Again, the advancement of this particular report, it was in 2021, which says that not only the synthetic cells can be formed inside the cell, inside the lab, but they can grow, they can automatically divide, they can replicate, they can they can complete or they can control the basic processes of life to make it inside the lab itself. And this one is the recent, as you can see the date being mentioned here, November 2023. It says the synthetic first eukaryotic yeast project unveils the cells with the 50% artificial DNA. This is a Saccharomyces cerevisiae which has been synthesized inside the lab in November. It's a science report which is being mentioned there. So they say that 50% artificial DNA. That means why 50% artificial DNA? How they come to know? They come to know through the process of genome annotation, genome identification. They have just deciphered for the each cells that they require only the 473 genes that are required to maintain the DNA, the life cycle of the DNA of an individual eukaryotic cell. So until unless we don't have the information of the function or the structure of the DNA, we don't have the annotations of these all molecules, we cannot go for these kind of research. We cannot create the life inside inside the lab. And why to create life inside the lab? Because morally or ethically, if you see that this is a controversial topic to comment on, but there are many ways these cells are required. They are things that can be done by these synthetic life forms which are created inside the lab. So with this, I'll be putting a halt to this lecture or interactive session. So this is from my side. Thank you, everyone.
Thank, thank you, madam. Thank you. It was very wonderful, and you need more time to show this through experimentation, because for this experiment, no need of any lab requirement. So I will take your time in next time, so that you can show all these software to the participants. Before starting the
Yes,
question
yes.
session, I welcome our Professor Rakesh Patel sir. He is as a participant, and you all know, Gujarati student all know about him. His book we all refer in microbiology practical. Rakesh Patel sir, are you listening sir? Welcome sir. Rakesh Patel sir. आज आई कैन सी आइकन हियर राकेश पटेल सर, आप हैं? राकेश पटेल सर का किताब हम लोग वो माइक्रोबायोलॉजी प्रैक्टिकल में जो गुजरात में उनका सर का चलता है सर.
Hello.
यस सर, वेलकम सर, गुड इवनिंग.
Good evening, Gopal Ji. Good
सर
evening,
आप
madam.
अपना कैमरा भी ऑन करिए सर, बच्चे तो आपको देखें, किताब तो देखा हुआ गुजराती बच्चे.
Camera is not functioning at present.
ओके ओके, सो नाउ प्लीज ask
Thank you,
your
Poonam Ji.
question if
Thank
you
you,
have
Poonam
any
Ji,
question, or give your feedback also. That will be a moral boosting for us.
Thank you, Rakesh sir. First of all, thank you, Rakesh sir, for joining this webinar or the
सर
session.
आप अपना फीडबैक, आई हैव सीन कि यू हैव जॉइंड एट द स्टार्टिंग एंड स्टिल यू आर हियर. हम बीच में स्पीकर को डिस्टर्ब नहीं करना चाह रहे थे, इसीलिए आपको मैंने वेलकम नहीं किया.
नहीं sir, ठीक है. But it was a highly informative talk, and at least for me, because I'm quite aged now, 65 plus, and therefore these subjects are even new to me. I gain a lot of information by listening to such a learned professors, and I am highly thankful to Gopalji for conducting such a series of lectures, which will add to the knowledge of even retired persons like us. Thank you very much.
थैंक यू सर, थैंक यू. दिस इज लाइक अ ब्लेसिंग. आई एम ऑलवेज ट्राइंग टू इम्प्रूव अवर ऑल नॉलेज पुल. जितना ही
Yes.
कोई 10 मिनट सुना, कोई 20 मिनट सुना, सो ओवरऑल नॉलेज पुल तो इंक्रीज हो ही रहा है. And with the help of learned person like Poonam Mishra madam and other speakers. So now question session. Please ask Harjot Kaur. Please ask your question, unmute and speak if you can speak. Hello. Harjot Kaur, yes sir. Harjot Kaur, you have question. अच्छा मैं पढ़ देता हूँ नहीं तो. She has written, for example, we have to annotate a single gene in tomato. Is it okay by doing domain prediction, motif analysis, phylogenetic analysis? Madam, are you listening? You can also
Okay.
see this chat.
Yes, yes. Yes, I'm just reading out the question. What she has asked whether we can annotate a single gene in a tomato by the domain production motif using phylogenetic analysis. So, Harjot, what I would suggest you, if you are trying to annotate the data, as I mentioned, that annotation is just not looking for the predicted motifs. You have to look. Annotation is complete incomplete until unless you are not looking for the structural component as well as the functional component. So you will have to look for the phylogeny. For that you have the phylogeny db, just an example. If you are looking for the other sequences, like for the which kind of protein it is behaving like, or it has the function, so you can pfam. If it is having signal peptide, you can go for signal p. For kind of interactive molecules are there, or to other proteins, so there are other databases which are interactome. So deciding for those interactome, so all those are required when you are looking for the annotation purpose. Because if you are annotating a naive protein or native a new protein, then what you will ultimately uh, try to figure out, or what you will try to do with it, ultimately you will want that the other persons who are also involved in such kind of research, they may get benefited, or they may get know that you have correctly done it. So you will submit it to the repository, which may be ncbi, which may be uniprot, swissprot, or other kind of pdb databases. You can submit it. So when you are submitting it there, they require the full annotation, not just the structural annotation that may contain motif predictions, or that may contain every other structural components. They may also require what putative function they may have. And here I would also would like to mention it, that not only the bioinformatic analysis, but you will have to, till the time you are predicting it, that is okay. But for making it a sure shot uh, annotation, what you will ultimately need, you will have to do it in the wet lab. You will have to identify the gene, you will have to mutate it, you will have to identify the function with it, you will have to go for the structure of that particular gene or the protein which is being formed from that protein. So you will have to look for all those kind of information when you are trying to annotate a single gene on the genome.
Okay, so C left, I think. Now, Sachin Rajput, you have raised your hand. Please unmute and speak. Sachin.
Yeah, thank you
Bolliye.
very much, sir, and thank you, ma'am. Very nice presentation. I want to know what is
Thank
this?
you.
Why should we do structural annotation? First question, and second question is, if I were to know that certain biochemical is produced by some microbe, for example, let's say penicillin or anything. So, how do I
You can
find
ask
out?
in
Basically,
Hindi also. If you are not comfortable in English, you can ask in Hindi. She can understand. She can reply you in Hindi. Anna.
Yes,
yeah,
no
Bolliye.
issues.
okay, that's all. That's all, ma'am. Second, how to identify a particular biochemical that is being produced by some microbe.
Okay, so answering to your first question, which is why need to annotate a DNA sequence or the DNA gene onto a genome? Why we need it? So why we need it? The simplest answer is, until unless, for example, until unless you don't have an idea from have just a simple question to you. Have you checked the DNA sequence or the nucleotide sequence of the protein or RNA sequence? Have you ever looked at these sequences, how they are being written
Yes, I have seen faster format
in the
basically.
faster format, in the simplest faster format? So when you are looking at, until unless you are not knowing the start codon, or the, you know, the protein sequence is what? The first amino acid which is present in the protein, it's a methionine. है ना? So until unless you are not knowing, if you are having the DNA sequence, then DNA sequence, it would be having the codons. That codon, maybe there would be multiple codons which would be coding for the methionine. Which codon is actually coding for the first methionine? How you are going to know? Until unless you don't know the position of the start codon on the DNA sequence, है ना? Or the sequence, or the DNA nucleotide sequence, till that time you don't know, है ना? So for getting the complete information, for that also you need to know what are the enhancers, what are the regulator sequences, how much the distance is from that. For example, in prokaryotic sequences, we know there are promoter sequences are there, TATA, T Tata box, Pribno box, promoter sequences are there. उनके बीच के distances भी हमें जेनेटिक एनालिसिस सॉरी मॉलिक्यूलर एनालिसिस से पता चलती है कि -10 है, -7 है. ऑल दीज इंफॉर्मेशन वी आर अवेलेबल विद. अगर हम ये सारी सीक्वेंस की पोजीशन को नहीं जानेंगे, these are the experiment which were carried out with the help of molecular biology, but now you have the software analysis which can shorten your time. You can predict the sequence which are there, how much distance is there, you can have the information. Then you should go for the structural annotation to make easy, or to make what I would say, to make it easier for you to carry out the research in that particular molecule, or the DNA molecule, or the proteins if you are looking for. Um, Coming to the next, what you are asking about, the which kind of my biochemical substance it is producing. So for that, what I would suggest, biochemical analysis is not just you have the biochemical, you just go for the protein. For that, you have to carry out a lot of experimentation initially itself. Which kind of biochemical it is? It is a kind of protein, or is it having some kind of fatty acids, or the fat molecules, or the lipid structures are there, or it's a kind of glycoprotein, it's a kind of carbohydrate residue. So all those characterization first you need to carry it out. And if you are having the protein, then you backtrace it, or you can look for the gene, or you can look for the protein, you can go for the blast sequence, you can identify what kind of functions this particular protein sequence is having. So even at that level, if you are trying to figure out the protein or DNA, so first thing you would need to have some alignment. Once you are having the alignment, you may get the sequence for which you can design the primers. You can go to DNA sequencing, get the DNA sequence, then only you can say you have to then clone it in the E. coli cells. If the protein function, you have to check the in vitro, in vivo study, you have to carry it out for checking the functional status of that particular DNA sequence. So I hope this might help you a bit.
Yes, ma'am. And in first question, you mean basic skeleton will be known by structural annotation.
Yeah.
All
Yes,
right.
basic
And in
skeleton. For
and
that,
in
you need to know
yeah, ma'am. Yeah, continue, please. Sorry.
basic skeleton would be known, and it would be easier. Why to start with doing everything which our ancestor scientists have done? You have so much information, utilize that, go it with the structural annotation. This is the first and foremost things in most of the cases the labs are doing now if they are looking for working at the molecular level. So that is what I think.
And ma'am, in second scenario, let's say I isolate as X microbe, and I just straight away sequenced its whole genome, and
Okay.
then I go blasting NCBI blast, right? And then
Okay.
it will give me blast region, basically aligned region. And in order to know what is the function of that region, we need to see that if that region which has been aligned, it has already been
Okay.
functionally annotated prior,
Okay.
then right.
What I would say, or I would suggest, just to know the function of a single gene, you don't go for genome sequencing. It's not a cup of cake for everyone to go for the genome sequencing for a single gene. Just for that, the simplest thing, DNA sequencings are available in different labs. You can go for the DNA sequencing for the single gene. In most of the cases, the genes are 1 kb, 2 kb. You can go for them. You can analyze it through the simplest method of the DNA sequencing. No, not required to go for the genome sequencing. Only then you will be getting it. Once you are having the DNA sequence, you just go for the gene NCBI or blast it for the 1 kb region. That will give you the initial hits by which you can get an idea that what kind of function can be associated with that particular DNA sequence, or what kind of structural relationships are there.
But if that region which has been aligned, if it is not annotated, then what would be the use of aligning?
If it is not annotated, or it is not already present earlier, then there are two ways. If you have listened, or if you have tried to listen carefully, the first one was the external evidence, and the second one is ab initio. Once you are not getting the hits from the already known databases, then you have to look for the what methods for the different known sequences, different known motifs, different known what I would say, the presence of the DNA sequences. If they are there, if they are not there, if there are repeat sequences, then you have to go for the ab initio method. Ab initio methods actually try to understand what is the difference between the external evidences or the homology based methods and the ab initio methods. So ab initio methods, you go for the alignment of those sequence, you go when the alignment between the two or multiple sequences less than 30%. Till the time it is having alignment more than the 30%, you can say the homology it works. And if you are not having alignment, even the alignment level is below the 30%, then you go for the ab initio, or you start predicting or looking for the structures which are available to denote it to the particular DNA sequence, and then say that this is the structure, or this is the DNA sequence which is present in other protein. Maybe it may be functioning
Okay.
as a same in this DNA sequence also.
So now, Sachin, if you need more information, and so you can connect with Poonam Madam one to one. I'm taking Sangeeta Pramanik question now. Madam, can you see in chat box Sangeeta Pramanik questions?
Just a minute, sir. For blast search, if we want to look for a ortholog gene in different species, but the genes how but the same genus. Acha, species alag ho, but for the same genus, how will we choose the best search result? We get a lot of result in that case. It's not characterized. Okay, when you look for those blast sequences, you may find the percentage of homology that is mentioned from the first with the highest one, and as you go down, the similarity percentage it will also go down. So it depends on, just see, when you are looking for the blast search, it's not something that you may not have some kind of hypothesis associated with your DNA sequence that you are looking for the blast search. You may have some kind of hypothesis for which you are looking for how your DNA sequence or the protein sequence it is working. Okay? So if it is not, if it gets multiple hits, the best possible hit which would be there, it would be on the topmost first line, and as you go down, it will decrease the percentage of the homology similarity. Okay na? So you can identify, you can correlate, and their functions are also being given there. For example, if a protein it is present in different species, for example a protein, take it like cut4 protein, that is a protein that I work upon. Cut4 protein, it is present in pombe, it is present in cerevisiae, it is present in candida. These are all present there. A unknown blast sequence is there. Unknown protein, the protein sequence I am putting it, I'm getting hits like 100%, 95%, 85%, 60%, 40, 30, 20. So in that case, if I'm looking for a protein which is having 100% fit with the protein of my interest or the DNA of my interest, that means the DNA or the protein which I am looking for, it is the same protein. 95, that means it may have some kind of orthology or the deviation in the other species, but the gene, since the genus is same, the evolution convergence is divergence is not that much in case of the differential species, so they may have some variations. And as you go down, you can look for the percentage identity and the how much it is correlating with the hypothesis what you are trying to predict with the DNA or the protein of your choice. I hope this might have solved your query.
Fine. Any other question? Sangeeta, are you here? Yeah, okay. Any other question?
Yes, sir. I want to ask that
Okay.
can
सर
synthetic
सर
cell be
राकेश
used
पटेल
for?
सर, आपका माइक अनम्यूट वो म्यूट कर लीजिए सर, वो बीच बीच में वो ये हो रहा है. यस डॉली, प्लीज कंटिन्यू.
Uh, good evening, ma'am. A very nice lecture delivered by you. I have a question that can we use synthetic cell as vector?
No. It is just at very primitive state. It's a we can use, but probably in the or cell is not taken as a vector. Vector is not a cell. What vector we take, it is a kind of DNA molecule. है ना? Plasmid, phasmid, cosmid, whatever we are taking, these are the vector DNA. Vector is not cell. We are transforming cells or just a simple organism, it's E. coli. We are transforming it with the vector DNA, having the heterologous gene in it, transforming it, then getting what we want to have have with that particular DNA or the protein. So as if now, if the synthetic cells are being produced, like in case of Cerevisiae, it's been produced very recently, or the mycoplasma for 15-20 years are being 15 years are almost done with that. So these are the research which are done keeping the future in mind. Right now they are not, they are at very primitive steps stages. They have to be developed, they have to be, because producing a single cell or synthetic cell is one thing. To with the go with the idea of taking the benefits in future, maybe that may help us, but these are not used right now as a vector or anything else at this step or stage, where these are being just produced to check out how these are performing, how these are maintaining itself, how these are going on. So that's it.
Okay.
Okay. So if there is no more question, then again, I'm grateful to Poonam madam for giving that 1.5 hour for uplifting the overall standard. And if anyone need more consultation, you can connect me or directly to Poonam madam. You can join on LinkedIn. I will tag her so you can connect with her in future also. That is also one of the purpose. One-to-one consultation you can also take depending upon her available time and her term and condition. 1 hour and 1.5 hour is only to initiate the things. You should understand the capabilities caliber of the speaker, and you can utilize her so that unutilized resource may be utilized properly, optimally. So she can give you time depending upon her free time. You can connect with her, anyone. So thank you, madam. Thank
Yeah,
you,
sure.
Rakesh Patel sir, for joining and boosting us. Everybody, thank you, and keep in touch. Again on 23rd November, there is lecture on RNA, non-coding, non-coding RNA and other RNA. So you must keep on attending. This is all free, but you must interact. You must read the topic before joining the lecture so that you can utilize it in better way. Keep in touch through Telegram, because sometimes I may not forward in different groups this information after few lectures. And also you can join my YouTube channel. This is reluctant, but I may add all these videos, taking permission from the speaker in future, not now. So don't say ki you are not joining and you will ask for that video lecture, so then you will not get. You must join, and then in future if you have missed, so I will add on YouTube. Fine. Any you can share your feedback, unmute and share your feedback if you wish to share, or you can write in chat box. Otherwise I will wind up. Anybody
Okay,
wish to
thank you,
share
Gopal sir.
anything? Ji madam.
Thank you, Gopal sir, and thank you, Rakesh Patel sir.
Thank you,
Since
ma'am.
I am a, yeah, though I am not a native to Gujarat, I have just shifted to Gujarat 3 or 4 years back. But no doubt my students are also going through with your books. So now I can also say that I am in touch. Or Rakesh Patel sir, I get connected with Rakesh Patel sir. So
Thank you,
that's an
madam.
honor for me also, for your presence. And Gopalji sir, thank you so much for giving me this platform uh,
Thank you.
to just deliver my bit to the students or to the faculties who may have joined here. So thank you so much, sir.
वो भी है, अभी
Thank you,
देखा
ma'am.
था रितेश पटेल भी, इज आल्सो फैकल्टी, शायद वो चले गए. रितेश पटेल आल्सो ज्वाइंट from Tolani College. He is HOD there.
Okay, sir.
Yes. So thank you everybody. Good night. We will meet in again on 23rd or before that also. So keep in touch. Thank you, sir.
Thank you. Thank you very much.
Thank you. Thank you, madam.
Thank you, sir.